that was a good exam, and actually it turns out that the grades are pretty good. Only I worry about a few folks who, it just so far of the 3 exams, folks who have been doing well continue to do well. The high score continue to be the same darn person. You find out who it is and ask him, it's a him. How he does it, right? Ask him, how do you do that? How do you continue to get the right answer? Okay? And again, it's in not only the material that you understand, but also in how you study, as well as how you read the question and answer written by me. Okay, so all of that is I hope that you will figure out. All right. We have 5 topics here. We only have 4 days to discuss. Does that mean I'm going to just zip through? No, I'm not going to just zip through. Well, sorry, yes, that's better. However, when things are truly reviewed, I'll just say that it's review, and then those slides I will zip through, but for the most part I will try to explain, because signaling is yet another difficult concept to understand. Okay? There won't be as much information, but the concept is actually more difficult. Okay? So all of this has to do with signaling. So we'll go on with review about the signaling systems, give you a little bit more detail, and then we'll talk about, I'll give you examples, 4 or 5 examples of signaling pathways that most people study, right? These are traditionally area signaling pathways that are well used by most cells in your body. And then we'll talk about big ideas, integration and hierarchy of regulation of signals, and then signal and structure, and then we'll put everything together in talking about cell movement. Okay? So that's what we're going to do. Hopefully we'll get to B today. So just a reminder about signaling system. You have a signaling cell and a target cell. Sometimes they're called the upstream cell and the downstream cell. Sometimes the sender and the receiver. Sometimes the inducer and responder. I use the terms, like I have been, signaling cell and target cell. A signaling cell has to talk to the target cell. And then we'll go through, and then we already went through the various signaling systems. But for this set of exam, we're more interested in the target cell. So far, we talked about the signaling systems, right, and adhesion and all of that. But now, the target cell, once it receives that signal, what does it do with it is what we're more interested in this set of exam. And roughly, this is what the signaling cell does: is that a signaling molecule binds to a receptor, activates it, which in turn activates a signal transduction pathway or an intracellular signaling. Or secondary cascade. There are multiple names for it. Okay, so there's a whole pathway that ultimately then will induce what we call effector functions, meaning that the signal would tell the cell what to do. And what are some of these effector functions? To regulate metabolism, to regulate gene expression, and to regulate whether or not the cell has to move or alter its shape, right? In terms of movement and altering shape, we're really talking about cytoskeletons. So now we're bringing back material that you already learned, right? Then basically this is talking about regulation of polymerization and the assembly and disassembly of cytoskeleton. Altered gene expression, we're talking about transcription factors, and regulation of transcription and translation. And metabolism, we don't talk about that much, right? That's more of glycolysis and all of these activities that you learn in other classes. So roughly, this happens in a target cell. Let's remind you about the signaling system. These are the signaling systems that we talk about: the paracrine system, endocrine system, synaptic, cytosolic exchange, and then juxtacrine system or contact-dependent signaling. Okay. For paracrine system, most of the signaling molecules are called, if you ever heard of growth factors, cytokines, some gases can be signaling molecules. Okay, and then autocrine system is of course a specialized paracrine system where the signaling cell, the target cell is also the signaling cell. Okay, when you talk about endocrine system, you hear mainly of hormones, but some growth factors can also be signaling molecules for in the endocrine system, for instance. And some hormones can actually function, albeit not very often, but some of them can possibly function within the paracrine system as well. Okay? Synaptic signaling, we really refer to the signaling molecules as neurotransmitters. So whenever you hear neurotransmitters, and endorphin, epinephrine, and all of that, they're all neurotransmitters. Some neurotransmitters can be glucose, can be a neurotransmitter. Some of them can be ions. Okay, so signaling cytosolic exchange the signaling molecule is usually ions, either ions like sodium, calcium ions, or sometimes glucose could sometimes be the signaling molecules. The thing about the last two signaling system is that they don't require a receptor, right? They do not require a receptor. So signaling for cytosolic exchange, we talked about this a little bit, has to do with concentration, right? And the juxtacrine system has to do with cell adhesion, either to another cell or to the extracellular matrix. Okay. Don't even attempt to memorize these slides. Just give you an idea of the plethora of hormones that are available. This is important. Okay. So now remember, we're talking about, we're paying more attention to the target cell, right? The target cell, how a target cell receives a signal is by having receptors. Where can receptors be found? They can be found in a number of different places, including inserted or embedded into the plasma membrane, so therefore the receptors on the surface. It could be a soluble receptor that is floating in the cytosol. Not floating, of course, you know that they're attached to the cytoskeleton somehow, but they're not embedded in a membrane. So receptors can be embedded into the plasma membrane or soluble in the cytosol, or they can be embedded into organelle membrane, but not on the outside. So for instance, there are receptors a lot of receptors are found in the ER and even nuclear membranes. Okay? We'll talk about one example, but most receptors, however, are plasma membrane bound that we know of. So the majority, 90-some% of all, if you take look at all receptors, most of them are plasma membrane bound. Okay. So in this class, the examples that we give, most examples that we give, except for this one we're about to do, are about plasma membrane bound receptors. Okay. The question is, why do you think most receptors are membrane bound, plasma membrane bound? What about the signal? Where the signal? But what does that say about the characteristic of a signal? Cell to cell. It's cell to cell, but what is the signal itself? What is the characteristic? That says, what you're saying is that these signals, most of them cannot cross the plasma membrane. Therefore, most signals are polar molecules. Okay? These are exceptions. These are non-polar molecules, but most signals are polar molecules. Therefore, they are stopped at the plasma membrane. Right? So the receptor must be plasma membrane bound. Whereas if you talk about intracellular receptors, then these signals have to be nonpolar before they can cross the plasma membrane, right? Because there are no endocytosis or transport proteins for signals. So if they need to cross the membrane, the plasma membrane, they must be they must do it by diffusion, not even facilitated diffusion. They must do it by diffusion across the membrane. Okay, and the only molecules that can cross the membrane are nonpolar, small nonpolar signals. So you were very close. Okay. Let's take a look at the first example of a signal that crosses that actually one signaling pathway, okay, and what the effective function might be. And these are hormones. Hormones are, for the most part, nonpolar or hydrophobic. And I think I've said this before. You don't find too many things that are absolutely nonpolar, right? Most molecules, at least organic molecules, are amphipathic. That means that they're mostly nonpolar, but they do have a small hydrophilic or polar residue. So these are hormones. And so hormone function mainly during the endocrine system. They are secreted by the signaling cell, go to the target cell. Once they're at the target cell, what happens? How does the target cell recognize that this is the I'm getting ahead of myself. Oh, all of this is just still review, right? This is review. Yeah, don't try to memorize all of these, by the way. These are just review. Let me go to the, I'm sorry, since I started, let me go to this. I just got ahead of myself, but that's okay. Let me go to this. So for a hormone that is nonpolar, that means that there is not a plasma membrane receptor. These hormones can actually, for instance, example would be testosterone, can actually diffuse across the plasma membrane, and the receptor is actually in the cytosol. Okay? And once the testosterone in this case binds to the receptor, the hormone receptor, now that activates. So whenever a signaling molecule binds to a receptor, that interaction will activate the receptor to do something. Okay? In this particular case, the hormone receptor can act as a transcription factor. The receptor itself is a transcription factor. So you ask, how can a transcription factor be activated? We'll talk about phosphorylation, we'll talk about proteolytic digestion, we'll talk about binding by, here's an example where an actual transcription factor is bound by a signaling molecule. So once bound by the signaling molecule, this whole complex gets transported into the nucleoplasm, and since the hormone receptor itself is a transcription factor, it has DNA binding site that it binds to a consensus sequence, right, or a response element to regulate transcription. For the most part, usually they actually activate transcription. Yes, ma'am.
Sorry, you said the complex goes into the nucleoplasm, and then what happens?
And then bind to the response elements to activate transcription. To regulate transcription, but normally, usually, it's an activating process. Okay, yes, sir.
Since most of these signals are polar and can't permeate through the membrane, is this pathway like a minority?
Yes. Yes, it is absolutely a minority. But the opposite is a lot, maybe not most, but a lot of signals in the endocrine system uses this pathway because there are a lot of lipid soluble hormones, right? If you ever take systemic physiology, you learn about lipid soluble versus water soluble hormones, and a lot of lipid soluble hormones use this pathway to activate gene expression. Okay. So this is quick and dirty, right? It's pretty quick. You don't really, there aren't any reactions to catalyze. Just bind to the transcription factor, and there you go. You regulate transcription. Easy enough, right? Okay. Since I got ahead of myself, let me draw back. Okay. So for others, okay, especially the context dependent. So we already talked about, we already talked about, let me, again, because I got ahead of myself, I want to make sure that everybody's on the same page. So a bunch of slides I passed over are just examples of signaling molecules, right? And talk about signals that we already talked about, synaptic signals versus paracrine and endocrine, right? So we are talking about those, and most of those are soluble polar molecules, right? Okay. Yes. Okay. So what about the last two signaling, the cytosolic exchange and juxtacrine or contact dependent signaling? How does that occur? And that is explained in this slide right here. Okay. So in terms of the cytosolic sharing, you have the gap junction, and in plants you have the plasmodesmata. Okay, and again, how one cell talks to the other is by one cell having an increase in certain ions mainly or molecules, that then these entities will move down the concentration gradient from one cell to the other. And by doing that, what happens to the other cell? Its concentration of whether that ion or molecules will get increased. And that's how they talk. And then the increase will tell the cell what to do. Example. These two are, this cell is activated to have a great increase in calcium ion in this particular cell, right? So it's told what to do. So it at the beginning is a target cell. Now, what does calcium ion do? And we'll look at more examples later on. Calcium ions can activate certain enzymes. So there you go. The signal, whatever it is, will tell the cell to perform a certain reaction because of an increased of calcium ion, which in turn activates certain enzymes, or is required as cofactor for certain activities. Now, the increase in calcium ions, calcium ions are not only used by this reaction, but also move down the concentration gradient to tell this cell to perform the same reaction. That's what it tells that cell to do, is perform that same reaction. Does that make sense? So that's how talking occurs, and that's how the signaling molecules and can be used. So that's why in this type of signaling that you don't have a receptor, because the calcium ion, even though we view it as the signaling molecule, but it, once in the target cell, is used directly as a cofactor for an enzyme, and it doesn't have to activate anything else. Does that make sense? Okay, so that's an example of the cytosolic exchange.
So you wouldn't consider that enzyme that it activates a receptor?
No. A receptor has a very specific, definition. And that's not it. That's a fair question to ask. That's a good and fair question to ask, but the quick answer is no. Okay. So how about contact dependent or juxtacrine? It means that the signaling molecule is either attached to the plasma membrane of the signaling cell, or the signaling molecule can actually be a part of the extracellular matrix, right? Either way, it's attached. It's not floating around. So therefore, the target cell has to come to the signaling molecule instead of the signaling molecule going to the cell. Does that make sense? So in this case, the target cell is what needs to move. and it needs to be moved toward the, at least in part, to the signaling. So this is contact dependent, meaning that the two cells have to be right next to each other. Okay? And guess what? The adhesion complexes that we talked about, all the anchoring junctions can also, not only are they anchoring junctions provide what? Flexibility and mechanical strength to the cell, to the tissue, but also can be used for signaling. See, so cadherin and cadherin, as well as integrin interacting with an extracellular molecule somehow. Okay, it can be used as signaling. So when we talk about cell migration, this comes back. These are just other examples that we won't talk about anymore, but these are important molecules during developmental biology. So Dr. Leonard Holm likes to put them in there to warn you ahead of time about developmental biology. Okay, so that's contact dependent. Okay, so we look at all of the, we reviewed all of the signaling systems, right? And then we talked about the signaling molecules itself. And now I gave examples, or two examples already. One is sharing, and the other one is the amphipathic hormones that can activate gene expression, right? So those are in the minority. We're about to approach examples that are the bulk. The bulk of receptors are plasma membrane bound. That was in the hand up with it. Let me see. Okay. Try not to get ahead of myself. So look at the transmembrane proteins or the plasma membrane bound. By definition, if a receptor is a membrane receptor, it must be a transmembrane protein, right? You won't have a receptor that is either an ectoprotein or endoprotein, right? It has to be transmembrane. Okay, so receptors that are membrane bound are transmembrane. Again, these are examples, and we're going to look at the G protein link receptor, specifically the enzyme link receptor, specifically other specialized receptors, and then later on when we talk about migration, we'll come back to A, which is integrin especially, but also cadherin are important in migration. Okay. So transmembrane proteins can be any of these proteins, and actually we'll talk about ion channel link receptor as well. Okay, so we'll take a look at all of these examples of transmembrane proteins. But again, the idea is that all membrane receptors are transmembrane proteins. So what about membrane receptors that are found in the nuclear membrane or ER membrane? Do they have to be transmembrane protein? And the answer is yes again. I already said all membrane proteins are transmembrane proteins. All membrane receptors are transmembrane proteins. I did not say all receptors that are plasma membrane bound are transmembrane proteins. I said all membrane receptors are transmembrane proteins, right? Regardless of which membrane. Okay? So that's a little tricky. Okay? So again, like I said, most signals are hydrophilic. Let's see. Okay. The idea of one cytosolic, always cytosolic, one non-cytosolic, always non-cytosolic applies here. How does it apply? It applies because you must have that, right? Once non-cytosolic, always non-cytosolic. What's in the non-cytosolic side, and why does it have to be there all the time? Well, what about the signaling molecule binding sites? You can't possibly have the signaling molecule binding site on the cytosolic side, right? It has to be on the non-cytosolic side. That's why once non-cytosolic, always non-cytosolic. The reverse is true, that The cytosolic domain then will be used to activate a signal, a pathway, signaling pathway. Okay, so that's one important thing about this slide, which you've seen in multiple times, but I talk about this slide about different things. The other important thing about this slide is the idea of linked binding. That means that if a signaling, how does the cell know that there's a signal outside here? Because the binding of the signal molecule binding site, right, will cause conformational change of that domain, which leads to the conformational change of the cytosolic domain, which then would perform a certain function. So both of those ideas are very important when we talk about receptors again. One is once cytosolic, always not cytosolic and the reverse. And the other important idea is linked binding. That if a cell, that's how the cell knows that the signaling molecules out here is by conformational change. That the non-cytosolic domain is linked to the cytosolic domain. Okay? Always keep that in mind. A receptor, and this is where Lance's question comes in, like a receptor can be, and a lot of them, most of them, or not most of them, a lot of them are themselves enzymes, right? So if a signaling molecule binds a receptor, then on the non-cytosolic side, then that causes the receptor to have catalytic, the catalytic domain of the receptor then can catalyze reaction. Or a signaling molecule can bind to the receptor. The receptor itself does not have an enzyme, does not have enzymatic activity, but it is linked to an enzyme directly. So in either case, we refer to these receptors as enzyme-linked receptors. That means that either the receptors themselves, receptors themselves are enzymes, or that they are directly linked to an enzyme. So once a signaling molecule binds to the receptor, an enzymatic activity will occur. Okay? And then we'll talk about the signaling intracellular signal transduction pathway. Examples, again, most of these examples are about developmental biology. Okay? Which you do take. It's a really interesting course. It's a hard, interesting course. I can't teach it because I don't know enough about it. Some receptors are ion channels. Okay? So there are different types of ion channels. Not all ion channels can act as a receptor. Very specific, very chosen, there are specific ion channels that are themselves receptors. Okay? If you ever took systemic physiology, these are known as gated ion channels. So whenever you hear gated ion channels, these are, for the most part, most of them are ion channel coupled receptors, or ion channels that are themselves receptors. By definition, what is a receptor? A receptor is a transmembrane, in this case, a transmembrane protein that can be activated by a signaling molecule. That's all it is. So these are ion channels that are usually closed, and then they are told to open by a signaling molecule. That's it. Okay. Once they are open, then ions are free to diffuse down the concentration gradient. That's the other thing. Right? Down the concentration gradient. So if you have something like a sodium-potassium ion pump, then those are really never a receptor, right? Those are transmembrane proteins, and they are transport proteins, but they're not receptors. Okay? Yes, sir.
Do we have any activated pump receptors?
Like I said, I can't think of any. All of the ion channel coupled receptors that I know of are channels. That's why they call it ion channel receptors instead of ion pump. So that it's called ion channel coupled receptor for a reason, because all of the ones I know of, at least, and I think that they're all ion channels that are receptors, but not pumps. That's a good, and that's the point I'm trying to make.
We're always going down the concentration.
And always going down the concentration gradient. Right? And yes, ma'am.
So the ions can never be the signal molecule.
I understand. So you can have that. Let's say, is this an actual case? I don't know, but I think it is. That if you have a build-up sequestration, right? So I was going to go into sequestration. If you have a build-up of, let's say, sodium ions out here, and then the build-up sodium ion, what does that tell the cell? That tells the cell that, hey, there's a lot of sodium ion here that needs to come in. So the sodium ion would bind to a sodium ion coupled receptor that would open and allow the sodium that is being built up out there to move in. So yes, it can be. Or that signal, of course, can be other molecule as well. But yes, there. So the ion itself can act itself as a signaling molecule. Yes, that's a very good question. Okay. And that's probably the last time that we talk about the ion channel coupled receptor. So. Again, I jumped the gun, so we talked about this, right? We talked about the fact that there are receptors that are inside the cell, and then signaling molecules are, for the most part, amphipathic hormones that can diffuse across the plasma membrane, and then get to the receptor, and then the receptor gets transported into the nucleus. That's why they're called nuclear receptors. Okay? And then we did that. Are we good so far? Most of this is review. So there are a few added stuff here and there, but most of this is a review. We're good? Let's put some of this together. And of course, some gases can be signaling molecules like nitric oxide and carbon monoxide. Let's put some of this in context. How's that? So let's say you have two cells. You have this cell and that cell. Which of these two cells, according to this diagram, which of these two cells is a signaling cell and which of these two cells is a target cell? What do you think? Is this a signaling cell or a target cell? Signaling. Is this a signaling cell or a target cell? The correct answer is it depends. It depends. So on the exam, you would have gotten that question incorrect, and I'm hoping that you get it correct now because this might show up on your exam. So it depends. Depends on what? Depends on the signal and depends on the effective function. If you're talking about, let's say, increasing the amount of calcium inside the cell, right? Then the signals are, these are the signals. Acetylcholine, stress can be a signal, right? Bradykinin is a molecule during the repair system, right? These then activate transmembrane receptors that then can tell the cell to increase the amount of calcium. So in this case, this is the target cell. Calcium then is used by a protein called eNOS, nitric oxide synthase, which then catalyzes a reaction that can produce nitric oxide, which is released. Nitric oxide can be used as a signaling molecule. So in this case, now this cell that was a target cell now becomes a signaling cell. To release nitric oxide, the nitric oxide then, which is a gas that can diffuse across the plasma membrane to bind to GC, guanidinoyl cyclase. Okay? In this case, that's an enzyme. That's not a receptor. That's an enzyme. Right? So then the nitric oxide can be used by GC to catalyze this synthesis or production of cyclic GMP. that then can tell this cell to perform these functions right here. And in this case then, this is a signaling cell, this is a target cell. In this case, this is the target cell and the signaling cell didn't show. Acetylcholine could be a neuron somewhere. Does that make sense? So signaling cell, the point of that exercise is for you to understand that the signaling cell and target cell, what determines the signaling cell, what determines the target cell has to do with the pathway. Has nothing to do with the individual cell. Is a neuron always a signaling cell? No. Neurons can receive information. When it's received information with that pathway, it is actually a target cell. Right? So again, there's no such thing as a definitive target cell, right? Epithelial cells are target cells. No, that's never going to be the case. Okay? Very important concept. And again, hint, it might show up on some exam in the future. All right.
Maybe.
Maybe. keep you guessing. Let's see. Okay. So now, we talked briefly about the receptors, right? What about, I keep talking about the signal transduction pathway or the second messenger cascade, which is different from a second messenger. A second messenger is different from a second messenger cascade. A second messenger cascade is a signal transduction pathway. Why do I give you all these terms? Because these are terms that you would read in different papers, in different textbooks, they would use different terms. So it's unfair that I will just have you learn just one term because you might miss the other terms if you read something else. Okay? So second messenger cascade, signal transduction pathway, intracellular signaling, they're all the same thing. They're all a series of events, reactions that occur. That occurs, which is activated by an activated receptor. Does that make sense? So a signaling molecule binds to the receptor to activate the receptor. The receptor in turn activate a signal transduction pathway. Okay? Signal transduction pathway can be extremely complex. And if there were 1,000 different signal transduction pathways, there are 1,000 different ways to do it. So there is no such thing as a definitive example of a signal transduction pathway because they're all different. So all we can do is talk about specific signal transduction pathways to give you an idea of how they're regulated, okay? And that's the best I can do for you, okay? So I've chosen four signal transduction pathways that have been well studied. Probably 2 or 3 Nobel Prize winners got their Nobel Prize because of the signal transduction pathway. So this is a big slide, right? It just describes, basically it's a complex sentence, it's a big sentence that says that signal transduction is complex. That's what that says, okay? So if you think about signal transduction pathway, it's not just a series of events or reactions, but what does each reaction represent? It represents some of these things right here, right? What does each reaction tell the cell to do along the way so that ultimately there is an effective function that occurs? It can tell the cell to, it can be a relay of information. So if I whisper something to Jehan and then she whispered down the line, that's relaying, right? And why the relay? Why is relay necessary? Because there's something here in the middle of the cell. Let's say, yes, there is something in the middle. Let's say the Golgi apparatus, right? The signal wants to tell the Golgi apparatus what to do. But the signal is out here. Golgi apparatus is here. So then the signal has to activate the receptor, which then activate a series of events to reach this Golgi apparatus. That's a rough explanation, right? So that's why you have a relay. How about a very, very fairly general, in fact, this is what happens most of the time, and it has to do with the characteristic of an enzyme. when we talk about amplification. How is amplification to amplify a signal? Is that connected to enzyme? What do you know about enzyme that would allow amplification? Anybody? Any taker? You've taken enough chemistry courses. What is a characteristic of an enzyme?
It lowers the activation energy.
That's
So
one.
for,
What's the other characteristic?
What was it?
Can be reused.
Oh, okay.
It's not always catabolic. It could be anabolic too, right? But it can be reused. So if you activate one enzyme and it can be reused, guess what? It can send out the signals repeatedly. That means that's amplification. Does that make sense? So I'm basically one of those enzymes right now that I'm getting the message across by teaching a class. So then I'm doing it repeatedly. So I'm amplifying the knowledge, right? Hopefully that's what I'm doing. I could be up here and talk nonsense. But that's amplification. So how an enzyme can carry out amplification is the fact that an enzyme can be reused. That's important. Okay? That's very important. And then how an enzyme can be reused is the regulation of activation and inactivation of an enzyme, which we already talked about. Right? You see all these things are connected, right? They're all integrated. Okay, so amplification is important. Integration, what does that mean? That means that a cell receives probably, there's a slide later on that a neuron can receive up to 100,000 signals at any given time. So imagine you stand in the middle of 100,000 people all screaming at you, telling you what to do. Sometimes opposite things. Sometimes things that are absolutely impossible for you to do, right? Then what you have to do is kind of take all of that, integrate everything that you hear, and then you can only do really one thing at a time, right? That's what integration means, is that you have information, lots of information. Which one is more important than which one? And then you integrate all these pathways. So it could be multiple signal transduction pathways that tell this particular protein what to do. Okay? The opposite occurs. One pathway can actually, one protein at some point can actually tell the cell to perform multiple reactions, carrying out multiple pathways. Okay, we'll have examples of all of this. Anchoring. When we talk about, let's say, anchorage of molecular motor, right? Molecular motor. So, or how can the molecular motor carry transport a protein complex by having the protein complex anchoring onto the molecular motor? How about the other type of anchorage is integrin. Integrin can anchor onto either intermediate filaments or microfilaments, right? So then there has to be a signal transition pathway that talk about anchorage. Okay? Modulate, that means that just regulate. Turning on, turning off, causing conformational changes. And then a lot, not all, but a lot, I dare even say most signal transduction pathway leads to gene expression. So gene expression would be an effective function of that particular signal transduction pathway. A bunch of gene expression then can tell the cell to perform a certain function. So effective function can occur at multiple points depending on what you're talking about. Are you talking about, let's say spreading, right? So from this one pathway, at this point, you may have 3 different effective functions right at this point. Effective functions, right. However, for this effective function, it continues to gene expression, right? So again, effective function is a term that is used. You can use the term effective function if you have identified the endpoint. Where's the endpoint that you're talking about? Is this the endpoint that you're talking about? If this is the endpoint that you're talking about, then amplification is the effective function.
Okay.
Does that make sense? Okay. So you hear me use the term effective function, effective protein a lot, and it's always, I always have to tell you where the endpoint is. what I'm talking about before I can use the term effective function or effective protein. Okay? Preform. This is important. Hardwiring. Preform. I already warned you that I talk about hardwiring. Right? Hardwiring meaning that everything is in place. Okay? So you can hardwire the fact that a receptor can actually be pre-bound to a scaffold protein that had already pre-bound to a series of enzymes, for instance. Why is hardwiring important? Because it allows for speed, efficiency, and accuracy. Right? So speed is, if you have everything hardwired, everything in place, then once a receptor is activated, then boom, 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 all these enzymes are activated in a series. So that's quick, because then you don't have to go out. Once enzyme 1 is activated, enzyme 2 is already there. The cell doesn't have to go out and find enzyme 2. So it's quick. It's efficient because, again, it's efficient. And then it's accurate because all of these things are already there. You have enzyme 2 and not enzyme 3. So it's accurate as well. Okay? So hardwiring is really important. Okay? Especially if you have a very long signal transduction pathway, then you need everything in place. In more detail, what does that really mean? This really means that you want the enzyme and the substrate to be in the same place. Even though enzyme 2 is an enzyme, but it acted as a substrate for enzyme 1, right? So here's a cascade or pathway right here. Okay, so again, hardwiring is important. Here's another example of the hardwiring. Is that you might not have all these enzymes in place until the receptor is activated. Once the receptor is activated, it actually then acts itself as a scaffolding protein to recruit all these proteins, right? Until all these proteins are recruited, the signal transduction pathway does not occur. So again, hardwiring, but in this case, the hardwiring only occurs after the receptor is activated. And this is hardwired beforehand. In either case, it's still hardwiring. And it's still, and in both cases, talks about the various function of a receptor. Yes, sir.
So I thought hardwiring was it's all in place.
Right.
So how is this hardwiring then still, since it has to recruit them?
Before the downstream signal occurs, are these all in place? Yes. There you go. We'll come back. We'll come back to switches. Okay. So one we've talked about, one we haven't talked about. So how to turn on and turn off enzymes. If hardwiring is so that you can have a signal transduction pathway occurring very fast, very efficiency, that means that the regulation of turning on, activating and inactivating will also have to be fairly quick, right? So how are enzymes activated and inactivated quickly? By a number of processes, including phosphorylation. And so phosphorylation and dephosphorylation is important. Phosphorylation, if I remind you, is the addition of a phosphate group. And dephosphorylation is the removal of that phosphate group. So add the phosphate group, remove the phosphate group. Okay? Phosphorylation is carried out by a protein kinase, which rips out which then they are coupled. Activity of a protein kinase is coupled with the hydrolysis of ATP. So ATP is hydrolyzed, then the phosphate group that is released then is used for phosphorylation. And then dephosphorylation is when that phosphate group is removed, which is carried out by protein phosphatase. This might look like phosphorylation always turn on and dephosphorylation always turn off an enzyme, but that's not the case. Okay? Phosphorylation may either turn on or turn off an enzyme, and dephosphorylation may either turn on or turn off. Okay. So be aware of that, right? So the best that we can say is that both phosphorylation and dephosphorylation regulate protein activity or enzyme activity. Now, is it true that most of the time phosphorylation turns on an enzyme? Yes, that's actually true, most of the time, but not always. Right? Is it true that most of the time dephosphorylation turns on an enzyme? And that's also true, but not always. Okay? So again, regulate. So that's easy, right? The idea of a G protein or GTPase. By definition, then there are a lot. By the way, G protein, GTPase. Never mind. Okay, so that's okay. It's what we call a superfamily. So there are a lot, hundreds, and even maybe thousands. No, hundreds of different G proteins or GTPases out there. Okay, they all have a similar means of regulation. How a G protein is turned on, how a G protein is turned off. Again, G protein, GTPase is the same thing. Okay? So GTPase is just GTP and then AIS, versus G protein is G and then protein. Okay? So by definition, a G protein is one that is inactive in off position when there is a GDP bound to it. So again, a G protein is one that is inactive when a GDP is bound to it, and is active when a GTP is bound to it. So again, a G protein is in its inactive form when a GDP is bound, and an active form when GTP is bound. How do you turn on or how do you activate a G protein? By carrying out an exchange, not phosphorylation, but an exchange. The G, in order to activate that G protein to form this, then that GDP is literally ripped out and replaced by a brand new GTP. We say this is an exchange. The GDP is exchanged for the GTP. Activation is not by adding a phosphate group to the existing GDP. Right? When does that happen? There's only one time in your cell that can happen. The production of GTP only occurs during one event. And if you've taken biochemistry, biochemists in here, I know that there are at least half a dozen biochemists in here. When you Krebs cycle, right? You form GTP that sometimes is converted into ATP, but GTP between succinate, that step right there. Right? Anyway, so that's the only time where you actually produce GTP. This is not one of it. So, this is not a phosphorylation event. This is an exchange event. Okay? Even when I say that a million times, some of you are going to try to force that in and say that GDP is phosphorylated here, and unfortunately I can't correct that. That's to turn on a G protein, therefore, is the GDP is exchanged for the GTP. To turn off a G protein, then the GTP is actually hydrolyzed, right? So is that the same thing as dephosphorylation? Yeah, kind of, sort of, dephosphorylation of GTP, but hydrolysis sounds better. Hydrolysis is more of a mechanism. So to turn off a G protein, GTP is hydrolyzed to form GDP. Okay? We'll talk about this multiple times. The power of amplification. The power of amplification is this. That, imagine that your eyes, right? It can receive one photon, and that one photon can activate 500 molecules of G protein, which then induces the synthesis of 10,000 cyclic GMP. Remember that slide that we showed that nitric oxide activate the synthesis of cyclic AMP? That's basically one nitric oxide molecule activating, a few, synthesis of a few cyclic AMP. In this case, one photon can activate the synthesis of 10,000 cyclic AMP. That's amplification. That's incredible, okay? And each cyclic AMP, of course, will be used for different reaction. Sequestration. Don't forget sequestration. Right? So during cyclotransaction pathway, we'll talk about regulation of enzyme, how to turn on, how to turn off enzymes. And sequestration, remember the various ways to regulate protein expression? Protein function, it could be allosteric binding, it could be phosphorylation, it could be proteolytic digestion, it could be sequestration, right? So calcium can be sequestered either in the extracellular matrix, in the ER lumen, or in the matrix of the mitochondrion. And then normally you don't have calcium in the cytosol, so in order to activate that a particular protein, then calcium has to leak into the cytosol, either within the ER, within the extracellular matrix, or sometimes, not very often, but from the matrix of the mitochondrion. Okay? So that's one way to regulate protein or enzyme activity during a cyclotransaction pathway. And again, this is just a review. Okay? Something that we haven't really talked about is calcium. Is calcium binding proteins. So sequestration of calcium is one way to regulate protein function. If you sequester calcium, then certain protein in the cytosol won't work. If calcium is released, then that protein works because calcium is used as a cofactor. Most of the time, calcium that is released into the cytosol doesn't diffuse toward a certain enzyme. Each calcium ion is actually delivered to a specific enzyme. Okay? And is delivered by a calcium ion binding protein. And there are about over 200 of them. So what does that mean? Why are there so many calcium ion binding protein is the question. Why do you think there are so many?
Specific, yeah.
Specific to this different function. What is specific to this different function? Enzymes, right? So each calcium binding protein will deliver the calcium ion to a specific enzyme that will carry out a specific effective function, and therefore, if each enzyme is cell-specific, then the calcium ion binding proteins are also cell-specific. Not all cells, each cell. You can't find a cell that would express all 200 or so calcium ion binding protein. It depends on its cell-specific. Now we're back to the pattern of gene expression. Okay. So far so good. Last slide. I'm where I want to be. So far, since I have a minute to spare, and I felt like at the beginning I tried to rush through to get through all of this. So I kind of got the slides out of order, but is everybody kind of set with where we are right now? That most of what I introduced today is either a review or setting you up so that we can talk about specific pathways. And then once we talk about specific pathways, then we'll talk about effective function. Okay? And at the very end, we'll use cell movement as that. So then the last thing is, what does the signaling pathways, what does cyclotransaction, what does one cell tell the other cell to do? Everything. Okay? So the idea is this. Two ideas roll into one. Number one, a cell won't do anything unless it's told to do so. Right? cell will not. I guarantee you that. The cell will not, even deaf, it will not do it until it's told to do so. So there has to be a signal for the cell to do anything, number one. One big idea. The second big idea is kind of sort of in with here, kind of sort of rolled in here, where if a cell, the cell is not only told to do something, but it's also told not to do something. If a cell is told to die, then it will also be told not to live. The cell can't just be told to die. Because what happens? It will try to die and then kind of try to live at the same time, and that's not efficient, right? So if a cell is told to die, it will be told not to live. If a cell is told to live, it will be told not to die. Sounds redundant, but it's not, right? A cell needs to have a clear-cut signal, a clear-cut command of what to do. So if it's told to do something, it will be told not to do the opposite. If it's told to divide, if it's told to divide, it will be told not to differentiate. If it's told to differentiate, it will be told not to divide. You would think that since cell differentiation and cell division are coupled, but still, if it's told to do one thing, it won't do the other. If the cell will be told to do one thing and be told not to do something else. Okay, that's very important. So we're actually going to pick up from this slide on